the last TorBUG of the season. Today we have Julian, who's a third-year undergraduate student. He's in the Bioinformatics and Computational Biology Specialist program here at U of T. And so he's going to be opening up for us and talking about BioRender, which is an interactive cell for the web. And following his talk, we'll hear from Julian. Yeah.
Alright, hi guys. I'm Julian. I'm a third-year student doing bioinformatics here at U of T. And for my third-year course project, I created this web software called BioRender. And so I'm going to show you guys today what BioRender is about and what it holds. So, BioRender at a glance is basically an interactive cell for the web. And what does that mean when I say an interactive cell? So it's a library written in JavaScript that allows you to create interactive cellular and molecular models. These models use PDB files. They are highly realistic. They are to proper scale, and they are fully interactive. You can go from the size of an entire cell down to a single nanometer micrometer. So it's powered by WebGL. And that's important because you can just go to it right on your browser, and it works on basically any modern computer. So the overall arching goals are to create a scaffold for developing high-quality educational material, a visual tool to work with existing and future biological data sets, and a stepping stone towards cellular simulation. So before going any further, I'm just going to jump into a quick demo. Not fishing one sec. Alright, so it's a bit wobbly right now. I'll go to this screen. So there are two camera modes. Okay, so right now we are inside the mitochondria. And so these are ATP synthases. You can see how, as a dimer, they pinch the membrane into this structure. We have the electron transport chain proteins all along the wall of this membrane. And I can go and take a far out zoom and you'll see the whole structure. And something I'm going to talk about is that this structure is actually procedurally generated. So the shape of it is using like random numbers, so you can sort of have different runs and different parameters to change how the mitochondria looks. And so yeah, this is about 1,500 nanometers long, and the membrane is 4 nanometers thick. And you can see how you have to bring models in and out of the scene, because otherwise it just overflows what the CPU, what the graphics card of everyday laptop can do. And so that way you have to get an efficient rendering. Okay, so that's basically the mitochondria. And the point is that this is like a concept of how to do the rest of the cell this way. Because the way this is created is that you specify a membrane and you specify which proteins are part of that membrane, and it will automatically cluster those proteins on top of that membrane without colliding in an efficient manner. So why did I create this tool? The problem we have right now is that the current tools used to teach biology have not followed the recent skyrocketing in technologies available. For example, WebGL has been available since about 2011, and nothing right now has taken advantage of that for this scale. There are single molecule viewers, but that is not the whole cell. So currently, tools we use to teach students biology are textbook illustrations, simple 2D pictures, and animations when possible. And these are great. These are highly good quality and highly realistic illustrations, but they're missing one crucial factor, and that's that they're not interactive. And being interactive is really important for teaching biology because biology is a complex 3D system that requires you to sort of look at it and understand it in its natural state, which is as a 3D structure, not as a simplified 2D diagram that can obstrue things behind each other and whatnot. So the way that I would approach this problem is to increase understanding and engagement of cellular and molecular structures through 3D interaction. And that 3D interaction is necessary to improve our current ability to teach these concepts. And furthermore, such a tool like this can also approach the problem of lacking biological literacy among everyday individuals, because something like this is sort of a game, may be more approachable to those people. So the parts that are interactive is because you're engaging directly with the environment and you feel like you're there. You're able to explore the parts of the biological system that you want to take a look at and that interest you at that moment. And so this just triggers your brain and you're actively thinking about what you're looking at. And the ability to experiment with variables. For example, if I move this protein, how will that affect the system? And you'll be able to witness that on a scale of large-scale structures such as the mitochondria or even the cell as a whole down the road. This is just a video of a point before where I had the spinning ATP synthase barrel. That wasn't the demo I just showed you, but it was at one portion. Just everything we got together didn't get fully completed. So I'll go a bit through the workflow of this and how it was made and how that will let other people make certain structures as well. The overall is that you have to model large items such as the mitochondria and the plasmic reticulum. On those models, you would then paint protein localization information, and I'll go into what that means. You can then bring in a list of proteins in each of their densities. And finally, the BioRender will take all of this and construct an interactive scene that can be distributed online. So step one is modeling a mitochondria. And so I did this with Blender, and it was actually done first by hand, as in like using the mouse and the screen. And because Blender is essentially just a GUI to a Python API that talks to C++ API, and every action you do, it tells you what the equal Python command is. You can essentially figure out how to script out your workflow. And so the reason that's important that these models are made through procedural scripts rather than by hand all the time is you can modify the parameters. And this means that you can have sets of parameters for different cell types, different stages of the cell life cycle, etc., which adds a lot of variability and applicability to different scenarios for the system. And as well, it's very reproducible, and you just run the script again. So the way that protein localization is transferred into this software is that we can paint materials onto the surface, which essentially means that groups of vertices are labeled as belonging to one section. And then BioRender can be told which proteins go on which areas, and therefore place out proteins on their on where they should belong properly. So you can see here that the red areas are the pinched region of the cristae, and that's where ATP synthase dimers would go to pinch in the cristae. The blue region is where ETC proteins would go. The green is the outer membrane, and then the white area is sort of everything left over in the inner membrane. So once you have those two things, you need proteins. And the way that we transfer protein transmembrane information is similarly, you can paint a material over the region which should belong inside the membrane. And then that way, BioRender can get the proper scale and position it properly in the membrane automatically all for you. And the final step is it takes all this information and constructs for you a WebGL scene. This is looking down the cristae pinched region. So a potential application that I want to explore in the future is integrating with the gene ontology, specifically their cellular component tree, which stores terms of annotated cellular structures. And you see an example here, cytoplasmic side and mitochondrial outer membrane. They're quite specific. And what's quite cool with this is that proteins themselves are also annotated to these IDs. And that way you can potentially create an entire scene programmatically using these annotations. So given a protein, find out where it resides and then construct that all. So that's my presentation. Thanks for listening, and if you guys have any questions. I want also a big shout out to Smart Biology. They're my advisor for this project and helped out a lot with helping how to use Blender, providing models and resources, and overall was a great help and necessary for me to succeed. So thanks a lot, and thanks for watching.
Thanks, Julian. Any questions?
Yeah.
So you switch off and on visibility of proteins as you, if you're closer or further distant. Have you thought of swapping in and out models at different granularity instead?
So how do you mean by granularity?
So like if it's far away, the protein is approximated by a sphere. If you come closer, it's a deformed sphere. If you come even closer, it starts getting the shape it has, and if you're at the full molecular detail, only if you're very, very close.
Yeah, so there's a lot of graphic stuff for that. It was really hard to sort of get this to work on laptop quality graphics, and that was sort of the existing method I have right now, where the further away from you are, it's a lower quality detail model. There are methods to sort of get convex subdivision, like approximations of models, but I haven't gone deep into that stuff. But that's definitely down the road. And also, instead of not showing it at all, you just have like a billboard picture of the protein when you're really far away, and that would still look good from far away, but also render fast.
How much code is involved in the mitochondria model?
So I think it's a few hundred lines in one main Python file, plus several like utility Python files for working with. It's really annoying working with Blender because you have to follow their API, and there's really tricky stuff where this action won't work unless you're in a certain 3D view, which is not possible in the code. So you have to sort of override where it thinks it is, and really edge cases that kind of annoying. But I've sort of created a bunch of scripts to ease the process. Yeah.
I may have missed it, but how did you define the scale of the mitochondria compared to the proteins?
So the base scale was 1 nanometer, and basically I know the scale, I know the width of the membrane, and I know the region of the proteins that should span the membrane. And so I can scale those proteins based upon scaling that region, and then everything else from that follows.
But the whole total size?
So the whole total size was got from literature, like values, yeah.
Great.
Thanks.
Thank Julian again.